Hello student, welcome to EPG Pathshala. I am Dr. Sushma Talegaonkar from Jamia Hamdard. Today we are going to talk on the module Introduction to Liposome from paper Novel Drug Delivery System 2. Learning objectives of today's talk are Introduction, Advantages and Types of Liposomes. Second, Components of Liposomes, Modification of Liposomes and Limitation of Liposomes. The liposomes are vesicular, spherical, colloidal particles which are composed of lipid bilayer encapsulating an aqueous core as shown in figure 1. This diagram is showing hydrophilic head groups and hydrophobic tail portion. Hydrophilic head, and inside there is an aqueous core which contains hydrophilic drug, and hydrophobic double layer mostly contains hydrophobic drug. So this is the beauty of this colloidal carrier system, which generally encapsulates both hydrophilic as well as hydrophobic drug. Liposomes were discovered by Bungham and co-workers about 40 years ago. Since then, liposomes have progressed commendably. They also called Bungham bodies. Liposomes are successful drug delivery system in which hydrophilic drugs can be encapsulated in aqueous core and the hydrophobic drugs can be dispersed in bilayer of the lipid that we already seen in figure. Thus, the liposomes are one of the most and very few drug delivery system in which drugs of both the nature hydrophilic and hydrophobic can be loaded in the same particle. Since the liposomes, they are commercially viable colloidal carrier system, one of the most important colloidal carrier system. Now let us see what are the advantages of liposomes. Liposomes are completely biodegradable and biocompatible as they are made up of biodegradable and biocompatible lipids. Liposomes do not trigger any immunogenic or toxic reaction in the body. That is, they are non-immunogenic and non-toxic. Most of the lipids that are used for the formulation of liposomes are natural and are easily available. Drugs of different nature can be loaded hydrophobic as well as hydrophilic and amphiphilic drugs. Combination of the drugs having different nature hydrophilic or hydrophobic can be administered through liposomes. Sensitive and non-target tissues are not exposed to the toxic drugs when drug-loaded liposomes are administered in the body. Drugs which are unstable or are prone to inactivation in certain conditions such as acidic pH of stomach remains protected and stable in liposomes until they reach in the target site. Encapsulated drug remains protected from the external environment also. Targeting of the potent and toxic drug to the target tissues or cells is possible. This can be achieved by attaching targeting ligands, that is active targeting, or through EPR effect, that is passive targeting. Since the drug is targeted to the specific site, wastage of the drug at non-target site is significantly reduced. And this results in the reduction in the dose of the drug which is to be administered, and hence the less toxic effect and more patient compliance. Now student, let us see how this liposome is going to be formed. The mechanism of liposome formation is based on the nature of the lipid, that is the main component of the liposome. Lipid molecules is amphiphilic in nature, that is it has a hydrophilic polar head and a hydrophobic tail of hydrocarbon chains. When these lipids molecules are exposed to an aqueous environment and some energy is dissipated into the system in form of agitation, extrusion, sonication or homogenization, the lipid molecule arrange themselves as closed bilayer structure, as shown in figure. This way, lipid molecules avoid the encounter of hydrophobic moiety with the aqueous molecule. The hydrophobic chains of the two layers remain facing each other, while the hydrophilic polar moieties stay in contact with water molecules. In this figure, we can clearly see the hydration of lipid film. As we are going to hydrate the lipid film, the multi-large liposome vesicles are formed. When we are applying these multilamellar vesicles for some energy like the sonication, then this large multilamellar vesicles, they are transferred to large unilamellar or maybe small unilamellar vesicles. While when these multilamellar large liposomes, when we are going for the extrusion mechanism, they are giving a very small uniform shape and small unilamellar liposomes as in B. And this figure is also showing how this bilayered structure of the lipids, they are arranging themselves, and when we are hydrating this bilayered structure, they are converting into the svelte structure, and after absorbing with the water, they are generally converted into the sealed compartment formed by a phospholipid bilayer. Now we will see modification of liposomes. Conventional liposomes can be modified also to serve other purposes such as targeting and to overcome the limitations of conventional liposomes, such as their early removal from the blood circulation. They are generally taken up by reticuloendothelial system because they are mostly they are hydrophobic in the nature, and most of the time they are having negative charge. So they cannot, after their administration, they cannot go to their proper destination. So the following are the common types of modified liposomes. Under this heading, the first modification in the liposome, that is stealth liposomes. The term stealth means cautious and surreptitious action or movement with a purpose to accomplish something which cannot be accomplished through usual route. Thus, liposomes are made stealth in order to increase their residence time in the blood circulation. Stealth liposomes are thus also known as long circulating liposomes. Conventional liposomes usually bind with opsonin proteins which are present in blood and are identified by cells of reticuloendothelial system as antigens or foreign bodies. Consequently, liposomes are taken up by RES and are removed from the blood circulation. Conventional liposomes are made long circulating by coating their surface with polyethylene glycol or PEG derivatives as shown in figure 5. In this figure, it is clearly we can see that the liposome's outer surface is covered by polyethylene glycol, which is one of the most important hydrophilic polymer in pharma industry, which is generally used to provide the hydrophilicity on the surface of colloidal carrier systems. This imparts hydrophilicity to the surface of liposome, hence they escape their uptake by the cells of RES, thus clearance of liposomes from blood decreases and circulation time increases. Now let us discuss the classification of liposomes. Liposomes are classified on the basis of structural parameters. They are classified as unilamellar vesicles, oligolamellar vesicles and multilamellar vesicles. Unilamellar vesicles, they are represented by ULV. They have only one bilayer of lipid membrane. They are further divided as small unilamellar vesicle SUV, size ranges between 20 to 40 nanometer. They are very small in size. Medium unilamellar vesicles MUV. These are larger and have size in range of 40 nanometer to 80 nanometer. Large unilamellar vesicles LUV. These are very large unilamellar liposomes having size in range of 100 nanometer to 1000 nanometer. oligolemular liposomes OLV. These liposomes are made up of 2 to 10 bilayers of lipids which surround a large volume of aqueous core. The size of the oligolemular liposome lies in the range of 100 nanometer to 1000 nanometer. Third type of liposome is multilamellar vesicles MLV. These liposomes are made up of several bilayers of lipid. The aqueous core can be divided into a number of compartments by these bilayers. Multilamellar vesicles may also be encapsulating several unilamellar liposomes. The size of these liposomes is greater than 500 nanometer. The classification of liposome is summarized in this table. The different classes of the liposomes, they are compared in this table, and the size of the liposome play a very important role in differentiation, and according to their size they can be used for the various types of delivery system. Components of liposomes. Essential components of liposomes are mainly the lipids and cholesterol. Lipids. Lipids form the main body of liposomes, that is the bilayer which surrounds the aqueous core. The typical characteristics of the lipid is their amphiphilic nature. Amphiphilic nature means the coexistence of both hydrophilic and hydrophobic moieties in the same molecule. A lipid molecule consists of a polar head which is hydrophilic in nature. This polar head is covalently attached to one or two hydrocarbon chains which form the hydrophobic tail part. The structure of a lipid molecule is depicted in this figure. Types of lipids. Following are common types of lipids that are used to formulate liposomes. First, phospholipids. Phospholipids are one of the most common lipids used for liposomes formation. Glycerol containing phospholipids constitute more than half of the total lipids present in the biological membrane. Phospholipids are derived from phosphatidic acid. Following are the different type of phospholipids. Phosphatidylcholine, lecithin, phosphatidylethanolamine, cephalin, phosphatidylglycerol, phosphatidylserine, and phosphatidylinositol. Now let us see the structure of phospholipids. Glycerol moiety forms the backbone of the phospholipid molecule. Hydroxyl group present at C3 is esterified to phosphoric acid. Hydroxyl group of C1 and C2, they are esterified with long chain fatty acids. These chains lead to the lipidic nature of the molecules. Remaining hydroxyl groups may further get esterified to various different alcohols such as choline, serine, ethanolamine, etc., leading to the formation of a different phospholipids. Second type of lipid is sphingolipids. Sphingolipids are made up of sphingosine, which forms the backbone of the molecule. The three components of sphingolipid molecules are a molecule of fatty acid, a molecule of sphingosine, a head group which can be anything from a simple alcohol such as choline to complex carbohydrates. Examples of sphingolipids include sphingomyelin and glycosphingolipids. The last type of lipid is synthetic lipids. Synthetic lipids, as the name suggests, are the lipids which are synthesized in laboratory. They are divided into two types: unsaturated synthetic phospholipids and saturated synthetic phospholipids. Examples of unsaturated synthetic phospholipids. First is DOPC, dioleol phosphatidylcholine, and DOPG, dioluol phosphatidylglycerol. Examples of saturated synthetic phospholipids are dipalmitoyl phosphatidylcholine DPPC, dipalmitoyl phosphatidylethanolamine DPPE, dipalmitoyl phosphatidylserine DPPS, dipalmitoyl phosphatidylglycerol DPPG, dipalmitoyl phosphatidic acid DPPA, and disteroyl phosphatidylcholine DSPC. Another important formulation component of liposome is cholesterol. Cholesterol or derivatives of cholesterol also forms an essential component of liposomes. The main function of cholesterol is to impart stability to the liposomes. It does so by interacting with the lipid bilayer of liposomes. Structure of cholesterol. Cholesterol has a hydroxyl group OH at third position, which forms a small polar head. The nonpolar tail is formed by the hydrocarbon chain, which is present at 17th position. The cholesterol too has an amphiphilic nature like lipids, which makes its interaction easy and strong. Function of cholesterol. Cholesterol performs following functions in the formation of liposomes. First, to stabilize the lipid membrane when liposomes are in fluids such as biological fluids like plasma. Second, to decrease the fluidity and microviscosity of lipid bilayer. Third, to reduce the permeability of the lipid bilayer towards water-soluble agents and hence to enhance the encapsulation efficiency. Mechanism of interaction of cholesterol with lipids. Cholesterol performs above-mentioned functions by acting as a motor for lipid bilayer and filling in the empty spaces that are present among the molecules of lipids. Thus, cholesterol fixes the molecules of lipid more strongly in the complete structure of the liposomes. Now let us see the importance of cholesterol. If the cholesterol is not included in the structure of the lipid bilayer, the liposomes would interact aggressively with plasma protein such as albumin and transferrin. These plasma protein have a tendency to extract the bulk lipids from the liposomes and consequently depleting the outer layer of liposomes, which will eventually result into the instability of liposomes. Because of its solubility properties and molecular shape, cholesterol significantly reduces these interactions between liposomal lipids and plasma proteins, thus imparting stability to the liposomes. Now, let us see another modification in liposome. Another type of modified liposome is magnetic liposomes. Magnetic liposomes are those which respond to external magnetic field. These are a type of external stimuli responsive liposomes. Magnetic liposomes are formulated by encapsulating a suitable magnetic responsive material such as iron oxide, magnetite and margimite is encapsulated in the core of the liposomes. In the presence of an external magnetic field, magnetic responsive material responds to it and moves inside the body as the magnetic field moves outside the body. This enables the targeting of liposomes to the cells or tissue where the action is desired. An area of application of magnetic liposomes is the treatment of cancer. Magnetic liposomes are introduced into the body and an external magnetic field is created around the tumor. The magnetic liposomes attracted by the external magnetic field reach the tumor and act on tumor mass specifically. Temperature responsive liposomes. Temperature responsive liposomes are another type of modified liposomes which respond to a change in temperature of the surrounding tissues, usually respond to a comparatively higher temperature. These liposomes are a type of internal stimuli responsive liposomes. Temperature sensitive liposomes can be formulated by using either lysolipids or synthetic polymers which are temperature sensitive, such as poly and isopropyl acrylamide. Temperature sensitive lipids are polymers sense the changes in temperature of surroundings and change their conformation. This results into disruption of the structure of the liposomal assembly and drug is released. Temperature responsive liposomes have been mainly explored for the treatment of cancers as cancer tissue bear a temperature slightly higher than the normal body temperature. Another type of modified liposome is pH sensitive liposomes. pH-sensitive liposomes are another type of internal stimuli responsive liposomes. These liposomes respond to a change in pH of different tissues of body and at a specific pH release the loaded drug. To impart pH responsive properties, these liposomes are formulated with some pH responsive lipids. pH responsive lipids undergo conformational changes at the specific pH. This leads to destabilization of the liposomal structure and thus the drug gets released into the surrounding tissue. Phosphatidyl ethanolamine is an example of pH-sensitive lipid. Phosphatidyl ethanolamine exhibits a change in the conformation at acidic pH. Immunoresponsive liposomes. Immunoresponsive liposomes are those which are formulated in such a way that they respond to the antigen present in the body or the diseased tissue. Immunoresponsive liposomes are prepared by attaching antibodies to the surface of conventional or stealth liposome. As shown in figure, these antibodies recognizes and attach to the specific receptor, that is antigen, present on the surface of the cell, such as cancer cells. This whole phenomena enable active tissue targeting through binding to the specific receptor of the cells. Another type of liposome is cationic liposomes. Conventional liposomes are either neutral or negatively charged. Cationic liposomes can be prepared by using cationic lipids for formulating liposomes. Cationic liposomes are formulated to enable the attachment of negatively charged targeting ligands such as hyaluronic acid. Dioctyl decyl dimethyl ammonium bromide is an example of cationic lipid. Now, another important modification in liposome, or we can say another important type of modified liposome, is targeting liposomes. Liposomes can be modified to target a specific tissue or organ by attaching targeting ligands on their surface. As shown in figure 7, the conventional liposomes, they are containing a particular ligand on their surface. Targeting ligands recognizes and bind to specific receptors which are present on the surface of target cells. These receptors are specifically present or are overexpressed on the surface of target tissues or the diseased tissue. Thus, the targeting liposomes having ligand on their surface recognize these target receptors and selectively bind to them, thereby sparing the other tissues of body. This helps in reducing the unwanted effects of the toxic drugs and reducing the dose to be administered. For example, hyaluronic acid present on the liposomal surface binds to CD44 receptors which are overexpressed in cancer cells, especially colon cancer. And folic acid can be attached to liposome to make them preferably bind to folate receptors overexpressed on the diseased tissues. This diagram is showing the interaction between the lipid and cellular membrane. In first case, the liposomes, they generally release the medicament and this medicament is going into the cell by the diffusion mechanism. Sometimes the liposomes, they can transfer their lipophilic compound to the cell. In another mechanism in which the liposomes, they are generally fused with cell membrane and after fusion they are releasing their content into the cell. In many cases, the liposomes, they can be totally internalized by the cell membrane and they can be taken up by the cell depending on their size and surface nature. Limitations of liposomes. Despite of several advantages, commercial success and utility, liposomes do suffer from few limitations also. Limitations of liposomes are usually unstable both physically and chemically. Physical instability: liposomes may fuse with each other to form larger vesicles or aggregates. Drug may leach out of the liposomes. Liposomes, they may have short half-lives. Chemical instability: ester bonds present in the lipids may get hydrolyzed. Oxidation of unsaturated acyl chain of the lipids may take place. Forms of physical instability. The physical instability may manifest itself in the following forms. Liposomes may fuse with each other to form larger sized vesicles or may aggregate to form lumps. This will affect their delivery to the target tissues and in vivo performance of liposomes, and thus will ultimately affect the therapeutic index of drug. On storage, drugs may leach out of the liposomes and move into surrounding media. This will result in the failure of purpose of the drug delivery system. Liposomes usually lose their stability when stored for a long time. Liposomes may have short half-lives. The problem of physical instability can be minimized by taking the following measures. Leakage or the fusion of liposomes can be a result of lattice defects of the membrane. This can be minimized by incubating the vesicles at a temperature above the membrane phase transition temperature to balance differences in packing density between opposite sides of the bilayers by transmembrane flip-flop. This process is called annealing. Second method, aggregation of neutral liposomes is a consequence of van der Waals interaction and is more prominent in larger vesicles. This problem can be overcome by imparting small negative charge to the lipids. Very small unilamellar vesicles tend to fuse because of relieving stress arising from the high curvature of the membrane. Including sufficient cholesterol in the membrane helps to overcome this problem. High amount of cholesterol also helps in minimizing the leakage of drugs from the liposomes. Lyophilization of the liposomes also helps in minimizing both physical and chemical instability by converting liposomes into solid anhydrous forms. The problem of chemical instability can be minimized by taking the following measures. The procedure which involves use of high temperature should be avoided. Second technique, preparation of liposomes should be carried out in the absence of oxygen in order to avoid the oxidation of lipids. The aqueous solution should be deoxygenated by using nitrogen. Liposomes should be stored in an inert atmosphere. An antioxidant agent should be added as a component of the lipid membrane. Poor encapsulation efficiency is another problem in liposome formulation. Encapsulation efficiency of liposomes is generally low, approximately 30%. To administer therapeutic doses, high amount of liposomes may have to be administered. But this will also expose the body tissues to very high concentrations of lipids. This may lead to nonlinear or saturable pharmacokinetics of liposomal formulation. Active loading techniques give better encapsulation efficiency. Uptake of liposome by RES is another problem in liposomal formulation. Though composition of lipids is similar to the composition of cellular membrane, the reticuloendothelial system of our body recognizes liposomes as foreign bodies. This RES uptake of liposomes is due to binding of opsonin protein which is present in blood and is recognized by the cells of RES. This causes the rapid elimination of liposomes from the blood circulation, which ultimately results in unsatisfactory therapeutic response. Uptake of liposomes by the cells of RES can be minimized by rendering the liposomal surface hydrophilic through coating it with some hydrophilic polymer such as polyethylene glycol or its derivatives. Sterilization of liposome. Since phospholipids are thermolabile compounds and are sensitive to heat, chemicals, and radiation, selecting a suitable method for sterilization of liposomes comes as a major challenge. Generally, liposomal formulations are sterilized by filtration through sterile membranes having pore size of 0.22 micron. But filtration method is not suitable for vesicles having size larger than 0.2 micron and also is not able to remove viruses. However, liposomes made up of thermostable phospholipids can be sterilized through autoclaving under certain conditions. This table is showing some commercially viable or marketed liposomal preparations. Mostly the preparations are for anti-cancer drug. M-11.Introduction is the first preparation which is containing amphotericin B. This preparation is given by the intravenous infusion. Manufacturer, they are Nexstar Pharmaceuticals, Gilead Sciences. Another commercially viable preparation is Donosomes. It is containing donorubicin and given by intravenous infusion. Doxil, another popular liposomal preparation containing doxorubicin given by the intravenous infusion. So students, let us summarize what we have discussed in today's lecture. First, liposomes are most successful nano drug delivery system or colloidal carrier system which are commercially available for the treatment of diseases like cancer. So many marketed preparations are available like Doxil and Biosomes. Liposomes offer several advantages such as biocompatibility and ease of loading both hydrophilic and hydrophobic drugs. But at the same time, liposomes suffer with few limitations also, such as poor encapsulation efficiency and their physical and chemical instability. In physical instability like aggregation, leaching out of drug, and chemical stability generally comprises of oxidation and hydrolysis of ester linkage. Third, measures need to be taken to improve the encapsulation efficiency and stability of liposomes, thus making them more valuable tool for drug delivery and other biomedical applications.